Okay, so to continue on with species interactions and talking a little bit about how they're actually figuring out that there's someone around them and whether or not they should enter into a conversation, so to speak. So the quorum sensing idea is looking at that. And when it was first discovered in Vibrio or Alivibrio fisheri, they thought, maybe it's just a one-off, it's unique to bioluminescence. And then they started looking more and more and they're like, oh no, this is like a thing. This is something that all kinds of microbes are doing. Initially, they were finding that gram-positives and gram-negative type bacteria did it a little differently. But since that initial, they've actually discovered more and more mechanisms in play in terms of what kinds of receptors, what kinds of chemicals. And so they're realizing there's a whole suite of options that microbes have in order to talk to each other and figure out who's around them, both their own cells, and now they're finding they can detect other species as well and communicate with other species. So we looked at this last time, realizing that gram negatives, typically, when they are doing their process, they're using a homoserine lactone of some sort. The side chain can vary. And they're producing it, they're excreting it out into the environment at low levels. So like as they're living their life, they're just doing this and it's at low levels. So it's not a huge cost to them. And then they, if you can imagine lots of these cells all doing that in a area close to each other, that signal, that chemical signal starts to accumulate. And then it will start to interact with the cells. And eventually that will get transduced all the way down to the DNA level and alter gene expression to allow them to do some kind of group behavior that gets them to accomplish a goal that they really couldn't do on their own. And I mentioned last time the idea of, if you become sick with Streptococcus pyogenes or something like that, if you think about the idea of warfare, how you want to build up a big army before you attack, that's kind of what's going on with that idea. So the homoserine lactones, the R is saying, yep, that side chain can vary in length. You have options. Different microbes have slightly different homoserine lactones. But in general, there's this like base similarity, and then the side chain alters. So that led a lot of scientists to wonder if there could be receptors or mechanisms where different species maybe or different genera of bacteria could understand each other's signals, even though they're slightly different. And so there's been a lot of study on that. And the short answer is yes, they can understand between interspecies. They're finding that more and more. So that also gets into the idea, of course, in a natural habitat, you don't just have like a bunch of E. coli hanging around. You have E. coli and lots of other species in that area typically. So we went through the idea of Vibrio fisheri, trying to show at the level of the DNA when, initially when a bacteria is just kind of hanging out in low densities, it's just, putting out that chemical at very low concentration. But if you can imagine lots of these cells all doing that, which the lower image is trying to show, then you have this idea of lots of the chemicals, they start to impact the cells and ultimately alter gene expression. And then here you can see they're trying to show that the gene expression is bioluminescence. Okay. Then with gram-positives, we saw it wasn't a homoserine lactone. It's peptide pheromones. And again, but the same idea, they're produced at low levels by the cells, they're excreted out. And then if you can imagine lots of these cells, then that signal does accumulate it triggers the shift into now they're going to start interacting with the cells, altering gene expression. So here's a couple of ideas of the peptide pheromones. They're just amino acids strung together in small chains, peptides. And so the more they looked, the more they realized this is really a thing. And more and more chemicals, besides what I've been showing you, there's been more popping up in terms of different chemical signals. But when you come down to it and you ask, why are they doing this? Well, there's lots of options, but basically it is a way for them to figure out how many of themselves, their species, is around, how many individuals are there. And so it's a way to sense their population, but it's also a way for them to engage in group behavior that allows them to do something beneficial compared to if they're all by themselves, they just couldn't do that and be successful. And so the idea of you getting sick, strep pyogenes, waiting until there's a lot of strep pyogenes in your body before they attack, because your immune system could easily wipe out a handful of strep pyogenes. But if they amass an army, then they could turn on all together. Let's turn on our virulence factors, let's attack. And when there's lots of us, and then we have a chance of surviving. So it is a way to also establish cooperation among a community of microbes. So we were looking at this last time. I think we ended off here with the idea that they're finding all kinds of, behaviors, the phenotypes, what's going on with these microbes and their ability to engage in this group behavior. And you can see there's a range of bacteria listed here, and this is by no means a complete list. And so the more they look, the more they find. So definitely it is allowing them to sense how many individuals of that species are around. But like I said, those chemicals like the peptide pheromones look similar to each other. The homoserine lactones look really similar to each other, even though they're slightly different with the side chain length. And so it really does make scientists think, are they able to understand different species, different genera, different organisms from different kingdoms even? And so this crosstalk is the kind of the short phrase that they use for this, is their crosstalk, or, looking at diverse species. And so they are finding, like I said, the short answer is yes. They're finding that interspecies communication can occur through this quorum sensing process. And it can even be from different domains of life. So for example, they know that plants and their associated bacteria and fungi can go through quorum sensing and can influence each other through that quorum sensing process. So the host and symbiont, that would be the example there with plants and their associated microbes. And so they're finding that humans also can enter into this process with microbes that are in you, and that quorum sensing is occurring. And so there are mechanisms in play where you could have wildly unrelated organisms, and yet they're able to communicate through this process of quorum sensing. So lots of excitement in this area of research, because one of the ideas, of course, when you think about antibiotic resistance, that we're running out of useful antibiotics to take if you're sick with a pathogen, they know that most human pathogens do quorum sensing. And so the idea is, could you come up with a mechanism to stop the quorum sensing? Because if you could somehow stop that line of communication, then they'd never know when to kick into the group behavior. It's like you're making them unable to see how many of, strep pyogenes, for example, are in your body. And so this idea of breaking up the communication is gaining a lot of research dollars are pouring in for that, because we're pretty worried about antibiotic resistance. They think if we come up with signals that can block quorum sensing, some kind of chemicals that can degrade the signal or somehow block it from occurring, that it wouldn't evolve resistance to that. The microbes wouldn't get resistant, and it would be an effective mechanism. So basically, if you got sick with strep pyogenes and you took a quorum sensing disruptor chemical, then even though you have strep pyogenes in you, because they couldn't quorum sense, they wouldn't know to engage in attacking you, and then your immune system could catch up and wipe them out. And so that's the idea. So there's a lot of research going into, they call it quenching, quorum sensing quenching. You can quench the signal. So lots of applications for this idea. Questions on quorum sensing? So microbes in you right now, they're talking to each other. And they're talking to you to some extent. Okay. So quorum sensing is a very out there signal, right? It's excreted out of the cells. Chemicals are out there. And so whoever's around can hear that, so to speak. And so because it's a very broadcasted signal. And so, anyone and everyone could hear that conversation because it's a chemical that's excreted out into the environment. So then this was discovered after quorum sensing, this nanotube idea. These are mechanisms that allow for cell-to-cell communication in a very private way. And so these nanotubes, this is one of the first papers published on it in 2011, when this was discovered. And so these nanotubes, it's pretty funky. What happens is that you have the cell membrane of a bacteria extending out into like a thin tunnel. And it will then make contact with another nearby cell and kind of fuse with their cell membrane. So it's an open tunnel then between the two cells. And so it's just cell membrane material and it's hollow. So they call the internal part a lumen, just like the internal part of your intestines is called a lumen. And then because it's just wide open like that, it allows for transport of different molecules and chemicals. So it's about 100 nanometers wide, which is wider than the pilus that's used in conjugation when we talked about plasmid transfer. And it's wider than that. So that has some implications for what can pass through the nanotube because it is definitely wider than a pilus. So that has led to a lot of speculation about what is actually getting passed back and forth potentially through the nanotubes. So tunnels between cells. So they're trying to show that here. And basically a cell will develop a nanotube and just kind of send it out there. And then if it finds a nearby cell, it will kind of latch onto it, so to speak, and fuse with that cell. And then they're trying to show some ideas of what could get passed. So they're showing plasmids as the black circles, and then the different colored balls are just some different molecules that can get passed through the tunnel. So they've been looking at this and asking the question, what can get transferred? What's the point? And definitely it's wide enough for small molecules to get through. So nutrients, for example, passing of nutrients from one cell to the next, maybe under starvation conditions, which they've done this in the lab and they deliberately put the microbes in a starvation condition and then they can see the nanotube formation and nutrient flow. But because of the wide diameter, they know that proteins, fairly large proteins can get passed, DNA can get passed, including plasmids. They've even seen viruses get passed this way from cell to cell. And toxins. So they have shown that experimentally, they can show that one bacteria can pass a toxin through the nanotube to a nearby bacteria that ends up killing that bacteria. And then they kind of suck away the nutrients for themselves. And so not such a nice interaction, but if you are in a starvation state and you're trying to stay alive, this would be a strategy to accomplish that goal. And they've also shown in lab studies that it can be a mutual thing where there can be passing back and forth. It's not one directional. It's just not from donor to recipient. You can have back and forth exchange through the nanotube. Or it can be a directed thing, like I said, where maybe you send out a toxin and kill the cell and then take advantage of that. So it's versatile in that regard, but it's definitely this idea that it's very private because nothing is out there in the general environment. It's all sequestered within the nanotube. So here's an actual picture under the microscope showing the nanotubes forming between cells. And so of course, one of the first questions that they came up with once they discovered it is this a thing that only happens among individuals of the same species? Like a bunch of E. coli are doing this, but they can't do it to another species or genera. And so definitely they were showing in pure culture, yes, nanotubes form among individuals of the same species. But then they started mixing cultures and growing them in mixtures and then looking to see what would happen. And definitely the answer was yes. And so here they did an experiment with Bacillus subtilis and E. coli, Escherichia coli, and they can show nanotube formation that way too. So interspecies between different species. So again, you know, in a community, in a natural habitat, you're going to have microbes that are in a mixed community. And this idea that they can have quorum sensing going on, they can do this nanotube formation. And so they can be very, very busy having all kinds of conversations through these processes. Some good, maybe some not so good in terms of attacking nearby cells with the nanotube idea, but I found this idea, nanotranslator. A nanotranslator opens cross-kingdom communication. So this is fans of the Hitchhiker's Guide to the Galaxy. You know that book? Okay. We'll remember the Babelfish, which fit within the ear and served as a universal translator. Although devices that could facilitate galaxy-wide communications remain fanciful, it may be possible to engineer devices that would allow living cells from different kingdoms of life on Earth to talk to each other. Progress towards such devices, blah, 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 in nanomedicine. They reported that they had built a nanoscale translating device that conveys chemical signals between members of two different kingdoms of life, namely bacteria and yeast. So they suggest that nanotranslators could be used to switch certain cellular processes on and off, or alter the activity of human immune cells to treat disease. So this idea of nanotranslators, nanotubes, they're getting excited about it because they're taking it and kind of running with it. How could we apply this to certain problems that are issues that we face? Okay, so nanotubes, funky idea, but apparently it's a real thing. Lots of bacteria are doing this. Here's another one. So actually this isn't unique. These vesicles, these extracellular vesicles, animals do this too, plants do this. So it's not unique to just microbes, but it's pretty easy to study these in microbes. So they've been looked at more carefully, I think, with microbes compared to maybe some of the other organisms. But basically what you're seeing in that picture are the bacteria are excreting these vesicles. And so a vesicle is just a spherical membrane bound compartment, essentially. All right. And so it is part of their cell membrane that they kind of pinch or butt off. And when they realize that all these bacteria can do this, and they seem to do it on a fairly frequent basis, then they started looking more carefully what's going on there. Are they empty? Are they full of stuff? And they find that, yeah, they're full of stuff, and they can have all kinds of things inside, including DNA and plasmids. SCFAs, those are short chain fatty acids. So Yeah, it depends on the microbe and the context of the situation. But they can have all kinds of different things inside. And basically it's just cell membrane, or if you're a gram-negative bacteria, it could be their outer membrane. That's part of their cell wall structure. But basically it's membrane material. And then they're finding in careful lab studies that these vesicles can find like nearby bacteria, and then they can kind of be fused with that bacteria. And so they can be kind of like a cargo delivery system, if you want to think of it that way, where you have this vesicle filled with maybe some nutrients or something, and it can get sent over to a nearby cell, fuse with that cell, and then the cargo can get released to that cell. They've even found in lab studies that it can be a way, they think, it seems anyways, that what's going on is the bacteria are using the vesicles to kind of distract viruses, phages, so that the phage attaches to the vesicle instead of to the actual cell. So they're kind of using it as a decoy to try to get away from being attacked by phages. That's what it looks like when they do these studies. So very interesting idea, really neat strategy if that really is a thing they're deliberately doing, which I would think is true. So these ideas are gaining interest. This is a paper that was published last year. And, you know, they do say here, it's not just bacteria, they're finding it in mammals, plants, and it's a way for communication to occur between organisms. And so they fit into the definition of a nanoparticle because they're so small. And they can have all kinds of metabolites, including DNA or RNA, and proteins, short-chain fatty acids, quorum sensing peptides. So this would be a way to be more private. If you encase your quorum sensing into a vesicle, that would be more private than just letting it all out there and the chemical just going out by itself. So they're looking into that idea. Is this a way for them to also be a little bit more private in their communication? Protect against bacteriophages, using them as a decoy. So they're interested in this. This particular paper is actually looking at if your gut microbiota are absorbing vesicles from, in this case, I think their main focus was plants, extracellular vesicles from plants. And they were thinking about if you ingest different herbs, for example, could some of that get transferred directly to the bacteria in your gut via vesicle transfer? So they're really getting into this and trying to figure out a little bit more of how these vesicles are being used, even as something as complex between, you know, the human host and their gut microbes. So here is another mock-up of what they're considering could be going on with these vesicles. And so some of this we've already talked about. You know, here you have toxins being wrapped up in the vesicle, decoy from the phage. Here's a virus attacking the vesicle instead of the cell, getting nucleic acids transferred. They're looking at it with plants as a host, as the microbes, looking to see if they can understand pathogen attack. Maybe the pathogens are doing a vesicle system to attack plants. Here they're looking at a more aggressive situation where you have attack again. Biofilms, this last image down on the right, looking at biofilm formation and dispersal. They think the extracellular vesicles might be involved in that as well. So this is pretty new research, but it is something that's gaining interest because now they're realizing, okay, you know, first we found out about quorum sensing, now nanotubes, now vesicles. So there's a lot of options microbes have in order to communicate with each other and assess their environment and their communities around them. And so way more sophisticated than initially thought. I remember when quorum sensing first came out, that was initially scoffed at. Scientists are like, no way, microbes aren't that sophisticated. And now, yes, they are. So it's an interesting development. So lots of interesting research going into this idea. Okay, so I wanted to introduce these topics so you can get a better feel for, these microbes. We've been talking all semester how these microbes are in these communities and they can be very complex communities. And okay, so now what? Yes, they are. I wanted to really emphasize they are very busy assessing their communities, talking to each other, coordinating their behavior, doing cooperation. So yes, there is competition. Yes, there is microbes, that are killing each other and doing all of that. You typically learn about that in a standard, ecology class, but you also need to think about the flip side, that there's a lot of beneficial group behavior and just a lot of communication going on a minute by minute basis. So it's way more sophisticated than initially thought. Questions on any of these ideas? Have you heard about any of these ideas anywhere else? I know you have. I talk about some of this in Gen Micro. Okay. All right, so is the next slide set posted in EDUCAT? Is there another one in there after this? Yes? Okay, thank you. Okay, so now that was kind of some broad topics on species interactions and processes in general. Now I want to start getting into looking at different biotic interactions more specifically. So I'm going to start with plants. So, and recognizing that this is a huge, huge field of knowledge and very active research. So obviously I'm picking and choosing what I discussed here with you. I'm breaking it down into kind of looking at a few different concepts. And the first concepts are thinking about the plant microbiome. And so we have, just like all the research going on right now with the human microbiome, lots and lots of grant money being thrown at that. It's amazing the findings they're coming out with human microbe interactions. Well, when all of that research started back in the early 2000s, that's when the granting agencies were really pushing, let's figure out what's going on with just the regular microbes that hang out with humans, and, not so much the pathogens, but all the others that just hang out with us on a day-to-day basis. And then the plant people said, well, wait a minute, this is probably happening with plants too. And so they started looking at that more carefully. So a lot of this research really took off in the early 2000s and working forward here. Although I have to say that, there have been some scientists that have been working on this for decades. And so, so there's earlier research too, but all of a sudden you see this flush of research coming out from the early 2000s. So let's talk about the outside first, the outside of the plant body. So epiphytic means on top of, epi, like your epidermis on the surface. And so epiphytic, anytime you see PHYT in a science word, think about plants, that's what it's referring to. So on plants. And lots of research here. When you think about the body of the plant, probably most of the research is looking at leaves, I would say, but realize that they have found microbes everywhere. Stems, leaves, flowers, fruits, surface of the roots too, of course. And so they are everywhere. If you stick to the above ground portion of the plant body, you hear terms like this, the phylloplane, the phyllosphere. These are areas, so the phyllosphere is a little bit broader term. The phylloplane is like right on the surface. You don't need to know the definitions of those. Just be aware that these terms mean you're talking about the really tight surface to, in this case, stems or leaves or flowers or fruits, for example. So if you're above ground and you're a microbe hanging out, let's say on the leaves, you can imagine that's a pretty volatile situation in terms of all the things that you could be exposed to. You're exposed to, really intense sunlight sometimes, then maybe abruptly getting shaded out, extremes in temperatures, rain, snow, sleet, all kinds of organisms coming along and eating the plant, and then you go into somebody's gut. So there's all kinds of situations that they have to deal with. So definitely a very kind of volatile situation, very changing environment. And so you're going to find microbes that are adapting to that. And in order to persist, if you want to persist, you got to deal with all of this. So one of the hallmarks, I guess, of being on the surface of above ground portions of the plant is you tend to see microbes that are pigmented. So both bacteria and yeasts, you tend to see like if you take some swabbing, I don't know if you notice this when you swab some leaves, but typically you tend to see pigmented, and it can be like Scott, my one grad student, when he would do some of his isolation off of apples, he was getting a lot of black pigmented yeasts, almost like they were producing melanin. And it is a pigment related to melanin. So very similar to the pigment like when you lay out in the sun and get a suntan or sunburn and you get that tanning, that's the melanin. And so it's the same basic idea. Your skin, the melanin starts to darken because it's absorbing that light energy and trying to keep you safe, rather than having that energy get to your DNA and proteins. And so they think that's the same mechanism, that the bacteria and yeast have pigments to deal with ultraviolet light coming from sun. And it's a way to protect themselves from that. And so when Scott was isolating all these bacteria and yeast off of his apples, he was getting all kinds of pigmentation showing up, black and pink and orange, and lots of interesting colors. Lots of yeasts show up. So yeasts are fungi, but they're single-celled. They look like big, plump, spherical, well, more oval. They tend to have an oval shape under the microscope. But they're much bigger than the standard bacterial cell for sure. And yeasts are well adapted to live on the surface of the plant. So you definitely have a nice community of those. They often have adaptations to get their spores accelerated out into the air. And so some of them have adapted mechanisms to kind of shoot their spores in order to try to get their spores to land maybe on a nearby plant. And so they have those kinds of interesting adaptations as well. And then if you're a microbe hanging out on the surface of a plant, you might want to be photosynthetic, take advantage of all that sunlight. And so you tend to see that too. If you do isolations off of leaves, for example, you'll find that you'll capture quite a few photosynthetic bacteria hanging out, having a nice kind of support substrate for them to get easy access to sunlight. And then leaves, well, yeah, probably the best would be, well, leaves, flowers, and fruits are pretty rich in sugars and sugar alcohols that get excreted by the tissues of the plant. And so again, you would find microbes that are well adapted to use those for their physiology. So if you're thinking about what kinds of microbes might live here, you have to think about the environment. You know, what's going on with the leaves or the flowers or the fruits? What do they provide or not provide? And how will that impact who can persist? They find that insects definitely play a role in getting transfer of microbes from leaf to leaf, for example, or plant to plant. You can get that all kinds of different insects. You know, I'm not going to go on and on, but certainly fruit flies, wasps, beetles, all kinds of different insects and pollinators too. You know, bees, for example, will be transporting microbes on their body. And so they're finding too that flowers are well colonized by microbes. And you can find, especially where there's pollen production and nectar production in the flower, you tend to have a lot of microbes hanging out there because it's usually really good nutrient source for the microbe. They can take advantage of that. And so again, yeasts really love this area. And so you'll definitely get a lot of yeast if you try to sample in flowers and look for microbes. And what they find, there's a lot of work going on with that, and they are finding these three genera of yeast, Candida, Torulopsis, and Rhodotorula, are commonly found. They're pretty broadly dispersed on different plants and in their flowers. So flowers are ephemeral, though. They don't persist really long. So then, of course, the scientists were like, okay, so we're finding all these microbes on flowers, but if everything goes okay, what's happening in the plant world is that flower gets pollinated, and then if everything goes okay with that, then the flower starts to shift into fruit formation. So if that's happening, then what? Are those bacteria toast? Are they going to die? Are they going to persist and get into colonizing the developing fruit? And so they've been following that. There's some really cool experiments showing that shift, and they can show that there's actually a succession of microbes that will show up on the fruit as it's forming. And typically you see the yeast dominating. They are like in first because they're really well adapted to both the flower and the developing fruit. And so they'll persist, but then they see that there are other microbes that tend to come in after the yeast are there for a while. And so as you get that flower to fruit formation, it's generally pretty gradual. There's a lot of change that has to happen as you get that fruit developing where the flower parts are. And so as the yeast are doing their thing, hanging out there on the developing fruit, you see that they're doing that autogenic succession idea where their processes, they're living their life, doing their physiology, it's altering the habitat a little bit, and it's letting other microbes then come in and get established. And so what they find in controlled studies is that as the yeasts are proliferating and really, they're happy on the fruit, they're getting enough sugar, they're reproducing, well, then the sugar concentration starts to go down enough where other microbes can come in, where it's a little easier for them to come on in and get established. And yeast tend to excrete fatty acids as part of their waste products, and we can see that bacteria that are adapted to use that to their advantage for their physiology and energy needs works. They've studied the idea that yeasts are excreting other things that they kind of just say, oh, they're growth factors. They're chemicals that stimulate the growth of other nearby microbes. And then you start to get this interaction where you get this community established and, they're using each other's waste products, so to speak, in order to take advantage of these chemicals getting excreted out. They've even done, a little off topic, but they've even done studies where they're looking at the flower what else the flower is producing besides the nectar and pollen, and what might be mixed into the nectar. And they're finding that caffeine is showing up in the nectar, which was like, okay, that's weird. Why would there be caffeine in nectar? And so then they started doing some nice studies on that, and they're showing that if the flower has caffeine in the nectar, the bees will visit those flowers more and remember that plant better. And so they keep coming back more and more. They think the caffeine is interacting with the bee and allowing them to have a better memory or a better map of where that plant is. And then when they go back to the hive, other bees will go to that plant as well. So, they do say that caffeine can help us with, our attention and our ability to concentrate and learn to some extent. And now they're seeing flowers are doing that in order to get the bees to come and pollinate. So pretty interesting dynamic there. I don't know if they've looked at caffeine and microbes. That would be interesting to see, but I don't know if they've done that yet. So, okay, so this picture is trying to get you to think about the size or the scaling issue here. So you think about plants, and I'm sure you've seen plenty of leaves that are very hairy, have all these lovely trichomes. The fancy word is trichomes that are coming off the surface of the leaf. Those are just long, skinny epidermal cells modified to be long and skinny and extend off the surface of the leaf. And a lot of plants that have this level of trichomes, that usually means it's adapted to more dry conditions, or areas where there's maybe some drought periods or maybe heat. This actually does help them to slow down their loss of water from their body. And so it is a mechanism they're employing. So not all leaves will be this hairy. Some don't seem to have much at all. But when you think about a microbe now on the surface, you can imagine that if they're on the surface of the leaf and they're surrounded by all these trichomes, it probably looks like skyscrapers to them, like you're in the middle of Chicago, downtown Chicago. And so that can have a huge impact on the microbe composition, their ability to colonize or to move off and get into different areas. Here are some other surface leaves, trichomes. But not all trichomes look the same too. When they look at under the, especially confocal or even electron microscope, you can see trichome design is beautifully varied. And so you can have some that are glandular, that have lots of essential oils inside, or they can just be long and skinny, or they can look more fat like this and more barrel shaped. So, but again, if you're a microbe, this probably looks like huge mountains, like Rocky Mountain National Park or something. So this extremely varied surface from a microbe's perspective, you need to appreciate that can create some very distinct microclimates and different patches of nutrients, for example. And so there's been a lot of effort trying to study all of this and figure out, like, where do microbes hang out? Here's another beautiful image. I believe this is a cannabis leaf surface with the glandular trichomes. And so those are filled with chemicals. And then you have some other more just spiky trichomes. And so again, from a microbe's perspective, they're so tiny, this has to be rather impressive to them. And then don't forget, if you've taken a botany class, you have to think about, there's also on leaves, there's pores, the stomata. And the stomata, from the plant's perspective, these pores or openings are very necessary to take in CO2 and do photosynthesis. And then that's their way of giving off water vapor too. That's how they allow water movement through their body, is they have water evaporating, and it literally sucks water through their body up in the roots of the leaves. So you have all these stomata, these pores, peppering the leaf. And now if you're a microbe, that'd be a great way to get in, right? If you want to get inside a plant, wait for the stomata to open. Here again, you're seeing the size difference of these microbes to the stomata. So this stomata over here is wide open, and you can see all these microbes, these little rod-shaped microbes on the surface of the leaf. Here, this stomata over here in the lower left is less open, but it's still open enough. You can see the microbes can easily get in. So this has been studied both for, can there be good microbes getting in there, bad microbes getting in there? And the answer is yes and yes. And so this is an excellent way if you want to get further in. This would be certainly a way to do it. In this case, this is Pseudomonas syringae. Yeah, it's one of the bad guys. All right, this one can cause disease. And so they were showing very nicely that this is one way it can get in and cause problems. Here's another one. So here you're seeing when they did this study, 1 times 10 to the 6th to 1 times 10 to the 7th cells per square centimeter. That's a lot of cells. So heavily colonized, they tend to show up at the base of trichomes or glands. They tend to show up in veined regions of the leaf. The red background is chlorophyll. The green and yellow are the bacteria. So they're well colonized. These are cotyledons. So this is a young seedling that just germinated, and the cotyledons are already well colonized. Okay, so we'll get into what are they doing next time. We'll stop there.